Hello, my name is Chloe, and I'm going to talk to you today about hierarchical Dirichlet regression for inferring relative cell village donor proliferation effects and simulation to improve experimental design. So cell villages are pooled populations of tens to hundreds of genetically diverse human stem cell lines of the same, typically of the same cell type, that can be used to investigate how genetic variation and environment influence cellular phenotypes. So you start a village experiment by obtaining deeply sequenced stem cell lines, and you can optionally differentiate these lines into different cell types. Then you take the individual lines and combine them into a single pooled population with donors in equal proportion and apply treatments and perturbations. Then you can perform omics assays and use low coverage DNA sequencing to impute donor representation over time. So experimental methods have not been optimized to ensure the reproducibility of these experiments and the adequate statistical power. Also, there's not really a lot of models that really leverage the complex data structure and the compositional nature of the data. So we want to address this by developing a hierarchical Dirichlet regression model for inferring treatment-specific donor and group proliferation effects from village time series compositional data. So here we have our effects that are outputted here, and we also estimate technical variation over time in the experiment. So the only inputs to the model that are needed are a compositional matrix that for each sample that includes the total donor proportions that sum to one of each donor or across all donors. And then you can include an optional design matrix that has sample and donor level predictors that include your treatments and different donor groups that you want to estimate effects for. So a little specific details on the model. Here we have our compositional matrix and we estimate these proliferation effects with a generalized linear model that employs the Dirichlet distribution to estimate this logits D, which accounts for the biological factors, the donor proliferation effects. And here we have an initial time zero compositional value for each donor per sample that is given to the model. And then based on across all these, we estimate a single beta per donor that is our proliferation effect. We can expand the model to include different treatment effects within this beta D value per donor, and also include these group effects such as sex, ancestry, genetics, disease status. Additionally, we have our overdispersion parameter that estimates replicate-specific dispersion over time. So we find that our model townlet performs much better and has higher sensitivity than other frequentist approaches. And these plots just show when we simulate villages with 10, 50, or 100 donors. And then additionally, we tested whether we can recover donors' treatment effects that we simulated, and we found that we can fairly accurately estimate treatment effects using townlet. So we found in our experiment that villages are reproducible and do capture cell intrinsic proliferation. So in tandem with our modeling, we performed various experiments to test. So we did a 12 donor village here where we estimated the village donor proliferation using our model townlet. And then we did a arrayed donor growth experiment where individuals were grown separately, and we used the exponential growth equation to estimate their or their proliferation. And we found strong correlation, even though these experiments were performed at different time points. Additionally, we did an experiment where we repeated a village using the same donors and found very strong correlation. Additionally, compositional modeling that accounts for overdispersion provides robust inference of village donor proliferation. So here's an experiment where we tested if individuals with the 16p microdeletion grow faster than controls. And here are the controls, and here are the 16p individuals relative to our baseline donor, which is All these growth proliferation effects will be estimated relative to a baseline, and we recommend choosing a median grower for this baseline, which our package will automatically help you choose. So here is our dispersion parameter theta, and as this value decreases, technical variation is increasing in the experiment. So we can see here that replicate 2 has slightly more variation than 1 and 3. And then finally, here is our 16p deletion proliferation effect, and this shows that the 16p individuals are indeed growing faster in this experiment than our controls. And then finally, we did another experiment where we tested for lead, and this is just demonstrating what is the output of our model, where we have per donor a lead effect relative to control proliferation, and the individuals with the diamonds have significantly different effects compared to the median. So these ones are growing slower and these are growing faster in the lead environment across 2 different lead doses. So for future directions, inference with our model is currently slow and computationally expensive due to the sampling. So right now we can kind of test a clearly defined set of hypotheses that we are basing on previous research findings, or we can use the proliferation effects to estimate a quantitative phenotype for downstream full genome-wide analysis using more efficient approaches. So in the future, we want to speed up inference so we can compositionally model proliferation effects across the full genome. So thank you for your attention. There is a preprint coming soon. Keep your eye out for it and contact us if you have any questions. Thanks to the Pimentel Lab and Wells Lab for all their support and my funding through the NSF and NIH. Thank you.